Welcome back to my course Aspects of Biochemical Engineering. Today is the last lecture. I shall make some summary and conclusion of the course and try to point out what is the purpose of this course, how we design the course, what are the different things we covered in this particular course. At the end, I shall discuss that what are the things you should get out of this course, and lastly I shall give the little introduction to my TAs, and also I want to show you some kind of video of 20-liter bioreactor for producing hydrogen. It is a continuous operation system. So let us start with that, how we design this course. If you look at this, that we divided into three different areas. Now, if you look, that first is we have the chemical reaction engineering. I told you to know the biochemical engineering, that first we should have some kind of idea on the chemical reaction engineering, and to know the chemical reaction engineering, first we try to discuss the chemical reaction thermodynamics. And we know the chemical reaction thermodynamics plays important role as per any chemical reaction is concerned, and from that reaction thermodynamics we can find out whether the nature of the reaction, whether it is spontaneous or non-spontaneous, and to what extent the reaction takes place. Then we discuss the chemical reaction kinetics, which appears to be the most important part of any kind of chemical process. To understand the chemical process, we want to find out different kinetic constant, rate equation we develop, try to do the simulation of the equation to find out the validity of the equation. And this equation, this particular kinetics we use for the reactor design, and in the reactor design is a very important part in the chemical engineering, because here we try to find out what is the volume of the reactor to get a desired amount of product. So we do the detailed analysis of the reactor. Now, after having this information, then we switch over to the biochemical reaction kinetics. In the biochemical reaction kinetics, first we try to understand what is the stoichiometry of the bioprocess, which is very important, because the reason is that if you know the stoichiometry of the bioprocess, then we can find out the material and energy analysis of the process, which is very important. From the stoichiometry we can find out what is the theoretical yield of the process, and so that we can design our process accordingly. And then after that we covered the enzymatic reaction kinetics, both by using free and immobilized enzyme. Now we know the free enzymes, when we have two type of enzymes. One is soluble enzymes and insoluble enzymes. And our substrate mostly is soluble in nature. So if your substrate is soluble, enzyme is soluble, then we consider as a homogeneous reaction. How the homogeneous reaction is, that reaction kinetics can be explained, that we discuss in this enzymatic reaction kinetics. But another situation that we have, or the heterogeneous reaction kinetics, where particularly we have immobilized cells. Immobilization of the cells take place on the solid matrix. As it is fixed on the solid matrix, then in the media, in the particular liquid, in the reaction mixture, you have two phases. One is liquid phase, another is solid phase. And we know in case of heterogeneous reaction, one phase, suppose until unless is come in contact with the other phase, the reaction cannot take place, and after reaction is over, then the product should diffuse to the previous phase, then we can get the product. So after having this information, then we try to understand the microbial growth, product formation, and substrate degradation kinetics. And if you look at the microbial system, we find out they follow the metabolic pathways, and in the metabolic pathways we have a chain reaction, and in the chain reaction we have number of steps involved. In each and every step usually governed by different enzymes, so it is a multi-enzyme system. So we try to, and we try to discuss what is the basic difference between the living system and the non-living system, and then we also discuss that what is the how we study the kinetics of the process. I told you that the major advantage of the biological process or biochemical process is that from the same substrate we can produce n number of products. I have given the example that sugar can be converted to acetic acid, sugar can be converted to citric acid, sugar can be converted to your acetic acid, sugar can be converted to lactic acid. So different products you can get, but if you look at the chemical process, as your product changes, their raw material changes. So that is and here in the biochemical process, what we do, we change the microorganism, and as we change the microorganism, we get the different type of products. So the living system main emphasis is to be given how we can allow our desired organism to grow in the reactor. So that we discuss in details in this, and how the microbial growth and product formation and substrate degradation can be taken into account in this system. After having all this information, then we switch over to the one important thing, that is bioprocess analysis, upstream and downstream processing. Now I told you that whenever we have any kind of bioprocess, that before whatever processes we involve before the bioprocess we call upstream processing, and after the bioprocess is over, then we shall have to do the purification of the process, that we call downstream processing. So here that in the bioprocess analysis, first we discuss the scale up and scale down of the bioreactor, which is very important, because in the day to when we do any kind of research in the laboratory, we develop the things in the lab scale, but lab scale it might be carried out either in the test tube or in the conical flux or in the very small reactor of capacity 2 to 10 liters. But that is not good enough for the commercialization of the process. But when you do the commercialization of the process, they will run in the very big scale. The main purpose of doing this is the environment that we have in the small reactor, similar environment should prevail in the big reactor. So for doing so, how the different operational parameter changes, that we discuss in the scale up of the bioreactors. And then there is another term very important, what we call scale down. Scale down basically, suppose we cannot do any kind of optimization study in the big scale fermenter, because if some negative results we get, then this incurred lot of loss in the fermentation process. So it is better we should do all the optimization study in the small scale reactor, and when we develop all the optimized parameter, then we can apply it in the bigger scale study, that we call scale down. Then after having this, then another very important thing is the process design. As per design is concerned, there is two type of design involved in the biochemical processes. One is called process design, another is called shop floor design. Process design basically involves that what are the parameters involved for operating the process, and shop floor design basically it is a mechanical design. So the scope of this particular course is only on the process design, not on the machine design or the shop floor design. So we try to discuss what are the parameters, how you can monitor different parameters for the process design. And after having this idea that we discuss the transport phenomena of the bioprocess, we try to discuss three different type of transport phenomena. One is momentum transfer, another is the heat transfer, and there is the mass transfer. That all the things plays very important role. As for example, momentum transfer, if you look at it is something similar to the fluid dynamics, that the mixing characteristics of the fluid plays very important role. And second, we have heat transfer that greatly involved for the sterilization of the bioreactor, because I told you sterilization that we shall have to allow our desired organism to grow in the media, and then naturally that all other contaminants should be free, so that our desired organism can grow. So so heat transfer is very important. Then I told mass transfer, because most of the fermentation process are operated aerobically, and major bottlenecks of the master that aerobic fermentation process is the dissolved oxygen concentration, because oxygen is sparingly soluble in the fermentation process. So mass transfer, how we can improve the mass transfer, that we discuss in this transport phenomena. Then after that we have two upstream processes. One is we have air and medium sterilization. Air sterilization, how we can do the air sterilization. I told you that two type of air is to be sterilized. One is called stagnant air, another is moving air. And suppose we spark the air through the bioreactor, that is the moving air. And stagnant air means, suppose we want to do some kind of operation in this particular room, then the room is to be sterilized, that is the stagnant air. And for the stagnant air sterilization, usually we use the UV rays or germicidal spray that sterilize this environment. But for the moving air, usually we use the physical separation technique. We use some kind of filtration technique just to remove the contaminants present in the air. And in case of medium sterilization, we find the heat is a good media for the medium sterilization, because water is good conductor of heat. So that we try to design both the air filter and medium sterilization process, that we discuss several numerical problems during covering this course. And then finally we come to the purification of the product, what you call downstream of this downstream processing, because whenever we produce any kind of product, that should be marketed in the purified form. And to purify the product, several steps are involved before the product is purified. So how the different this downstream processing can be operated, different filter, solid, liquid separation process, liquid the extraction process, how the crystallization process, the evaporation process, all we try to explain in this downstream processing. And finally we did this material and economic analysis of the process to give you idea how you can, whatever knowledge you acquired during this course, how you can apply it in real fermentation process. So this is the way we have designed the whole course. I hope it will be very useful those who are really working with the biochemical processes. Now if you look at the course content, that we started with this microbiology, then we started with microbiology, that we try to understand what are the different microorganisms present, and what is the classification, how their characteristics differ from each other, how they look under the microscope, how morphologically they differ from each other, then what is how their size varies from each other. So all these things we try to discuss in the microbiology lecture. After that we try to give you some information on the biochemistry of the process, because that is I told you that all the organisms, they follow the metabolic pathway. In the metabolic pathway you have chain reaction, you have to find out that reaction, so that you can have your strategy how we can get your desired product. I can give the example, suppose A to B, B to C, C to D, like this you are doing. So you are interested in the product of C. So the enzyme that is involved here, so this is, suppose this is E1, this is E2, and this is E3. So we shall have to put some kind of inhibitor here, so that the formation of D can be reduced, so that more accumulation of C take place in this process. So that so biochemistry of the process is very important. And then we find out that we try to discuss what are the different bioproducts that is available in the market and their market values, because that is very important, because those who are involved with the biotechnology or biochemical area processes, they should know what are the different products that are available in the market. And we try to classify that these products in the three different types: the low value, high volume products, medium value medium volume products, and high value high value and low volume products. So this is how we do the classification. After that, we try to discuss the stoichiometry of the biochemical process. I told you it is very important to do the material and energy analysis of the process. And after having this we started with chemical reaction thermodynamics. We try to discuss how thermodynamics plays important role in the chemical processes. Now after this we covered this kinetics of the homogeneous reaction, chemical reaction, how there are different type of reactions. As for example, we have irreversible reaction, we have reversible reaction, we have chain reaction, we have autocatalytic reaction. The different type of reaction, how this can be we can write the mathematical expression for this type of equation, how you can find out the rate constant, how you can find out the order of reaction. This is very important. I told you that the rate constant and order of reaction all are experimental parameter. With the help of experimental study, you can find out all these values. And after getting all the information, then we go for the we try to give you the information what are the different type of reactors are available. And reactors basically is a vessel in which the reaction take place. And then after getting this idea, we discuss that reactor analysis, and reactor analysis is very important. I told you just to find out that what is the volume of the reactor. Not only the volume of the reactor, we can select what type of process will be most suitable for your process. I can give the example. Suppose we want to produce a certain amount of product. Suppose I want to produce 1 ton of product per day. Now getting 1 ton of product per day, what will be the minimum size of the reactor? Because as the size increases, the involvement of the price for this process will be more. That is to be taken into account. So that is why the reactor analysis plays very important role. And I told you that two type of process. It's mostly we have batch process, we have continuous process. Another process we have what we call feed batch process. So 3 different processes we discuss in details. After getting the information of the chemical reaction engineering, then we started discussing the kinetics of enzyme catalysis reaction by using free enzyme systems. We try to discuss that what do you understand by enzymes. Enzymes basically they are the globular protein. Not necessarily enzymes are protein. Due to the advancement of organic chemistry, now it is possible to produce some enzyme that synthetically, but those number of those enzymes are very few. Basically the enzymes are protein and they are globular in structure. And the protein with active site, we call it that enzyme. So and purpose of the enzymes is just like a catalyst, and role of a catalyst, as you know, to lower the activation energy. As we lower the activation energy, the rate of reaction increases, but after the reaction is over, it remains unaltered. So all the different type of enzymatic reaction we try to discuss. Then we discuss the Michaelis-Menten equation, which is used for explaining the enzymatic reaction kinetics, and how later on that how Briggs and Helen they justify the Michaelis-Menten equation with the help of reaction kinetics. We try to discuss the different type of inhibition process. We discuss competitive inhibition. We discuss the non-competitive inhibition. We discuss the uncompetitive inhibition. Because on the basis of the inhibition, it is possible to estimate different type of chemicals present in the reaction mixture, particularly complex chemicals. I had given the example of pesticide, which is very complex chemical that can be estimated with the help of the enzymatic reaction kinetics. Now after having all this information, then we try to discuss the immobilized enzyme system. Immobilized enzyme system is advantageous as compared to free enzyme system. The reason is that in the free enzyme system, after reaction is over, enzyme remain as the impurities in the reaction mixture, so you have to take it out. but in case of immobilized enzyme system, you can reuse the enzyme again and again, and after the reaction is when your product is coming out, this is more or less free from the enzyme, so your purification process will be little bit simpler. So then we try to discuss the kinetics of the immobilized enzyme system, how the heterogeneous reaction kinetics can be explained. We have come across two new terms. One is called damped Kohler number and the effectiveness factor, and we try to find out how these two parameters that changes during the immobilization system. And I told you that two type of things that happen in the heterogeneous reaction kinetics, either is mass transfer control or reaction control. Now if you have mass transfer control, that means we shall have to improve the mass transfer of the process so that we can get maximum amount of product. If it is the reaction control, you have to improve the characteristics of the reactions so that we can get the maximum amount of product. Now after that we switch over to the the cell growth kinetics, substrate utilization, product formation, and that biomass formation, which is the heart of this biochemical engineering. So we have discussed this. We have in the several lectures we discussed different type of numerical problems. One thing that is very important here, I want to emphasize that when you use any kind of living system, we shall have to be very careful that about the age of the culture. Because why the age of the culture? If you look at the life cycle of the cells, we have lag phase, we have log phase, stationary phase, and the death phase. Every phase has its significance, because if you look at lag phase, it is known as the acclimatization phase, log phase is the active phase, stationary phase is the starvation phase, and death phase is the death of mostly the death of the cells occur. So if you look at that the organisms where it is very active, that is in the log phase. And usually that when you do any kind of inoculation of the organisms in the production media or in the inoculum fermenter, we should have to know that mid log phase and the late log phase. Now age of the culture, if it is in between the mid log phase and late log phase, you will get very good results. If you inoculate the culture close to the stationary phase, you may not get the appropriate results, your desired results in the fermentation process. So this plays very important role. And then we discuss this Monod equation, which is Monod model, which is something similar as compared to the Michaelis-Menten equation. Then we have come across with different type of other models like part equation. We discussed the Ludeking-Piret model. Part equation mostly deals with the maintenance of the cells, and Ludeking-Piret model discuss how the product formation that relate with the growth of the cells. And then there are other equations we have with the inhibition of the cells. We have here also we discuss the competitive inhibition, non-competitive inhibition of the process that we also may occur in the microbial system. So all these things we try to incorporate in this particular lectures. Now after having all this information, then we switch over to the process design, the design and analysis of the activated sludge process, that we have taken this as the instance that how when you talk a particular process, how the process design can be done, how the different operational parameters can be monitored or calculated. That we try to discuss in this lecture. We have taken into consideration activated sludge process, which mostly used for the treatment of the wastewater, and also we discuss the anaerobic digester, where we can convert the waste to some useful products, some kind of methane or hydrogen, which can be used as a source of energy. After having all this information, then we switch over to the scale up of the bioreactor. I told you this is very important, because whatever we develop in the small scale, that directly we cannot apply to the bigger scale until unless you have some kind of analysis of the process. The one analysis that is very much required is the scale up of the process. How during the scale up of the process we take into account that whatever environment under the environment you get the for your maximum product formation, similar environment you have to maintain in the bigger scale of fermentation process. So that you have to for doing so, how the operational parameter changes, that we discuss in this particular lectures. And then here we also taken into consideration, I told you that scale down of the process, that how suppose some high bulk with citric acid industry, now suppose we find out some parameters plays important role, has some significant role to increase the productivity of the process, but directly we cannot apply to the big fermenter. We first give the trial in the small fermenter, and if we are satisfied, then and only then we can go for the bigger fermentation process. And after this we try to discuss the different type of transport phenomena of the bioprocess. I told you 3 different type of transport plays very important role. One is the momentum transfer, another the heat transfer, another is the mass transfer. So we discuss all these things in details. Now we also discussed the air sterilization and medium sterilization of this process in details, how these processes can be designed. And then operation, we have given the instance of the operation of industrial fermentation process. I have given the instance of citric acid industry, and how you can do the material analysis of the process, how you can calculate how much material is required in different through the operation of the different process. As for example, how much cane molasses is required for getting a desired amount of citric acid, that how you can calculate how much lime is required for the precipitation of citric acid. So all these things we try to show you here. Then process control is we know that slowly step into the automation process, and as we switch over to the automation of the process, then we put lot of control devices, and how different control devices are there. And the biochemical process, couple of controls plays very important role, as for example pH, temperature, agitation speed, foam control. So that all these controls plays a very important role. Then we have downstream processing. We discussed the different units of the downstream processing, like filtration process, like different solid liquid separation process. Then we have chromatographic process, crystallization process, vaporization process, adsorption process. All these things we try to discuss in the downstream processing. And finally, we discuss the economic analysis of the bioprocess, just to give you an idea of what are the, because we have come across 2 type of expenditure that is involved in the bioprocess or any kind of chemical or biochemical industry. One is the fixed expenditure, and there is the operating expenditure. The fixed expenditure, how you calculate, and operating and expenditure, how we calculate, and that we compare with our selling of the product, what is the price revenue we get after selling the product. We try to compare with that, and from that we can calculate the profit you gain in a particular industry. So this all information we have given. Now after that, I recommend you a couple of books. You just go through this. I must say that this book is very good. This is the Biochemical Engineering for the Fundamentals, written by Bailey and Ollis. Then Chemical Reaction Engineering by Octave Lebensfeld. Bioprocess Engineering Principles, the Dudon, and Bioprocess Basic Concept, that is Suller and Karge. So these are the different books you can consult to clear your concept on this process. Now total assignment of this project, this particular course will be 12, and one assignment for 5 lectures. So that will be given to you, and this is the outcome of this particular process. To understand the chemical reaction engineering, that you should understand, I hope you have some idea on this, what do you mean by chemical reaction engineering. To find out the stoichiometry of biochemical process, so you get the idea how you can do the stoichiometry of the bioprocess. To understand the enzymatic reaction kinetics using free and immobilized enzyme, and then to find out the cell growth kinetic, product formation and substrate degradation, to determine the parameters involved for the bioprocess design, parameters involved for scale up of the bioreactor, to understand momentum, energy, mass transport involved in bioprocesses, to do material analysis of industrial fermentation process, to design air and medium sterilization, to analyze the downstream processes and also economic of the bioprocess. These are the outcome of this particular course. And I have two teaching assistant for this course. One is that Jhansi L. Banarasi. She is a very active person and she is doing PhD at the Department of Biotechnology. If you have any question, and her background is mostly on biotechnology. And we have Chandan Mahato. His background is chemical engineering. So if you have any question, you can in the forum you can raise that question. They will answer your question and to clear your doubts. And this is some I want to show you some of my published book. This is the biohydrogen production fundamentals and technology advance. This is published by CRC Press. Another is biohydrogen fuel for the future. This is the first book in the world we have published. This is published by PANS Transport Publishing Company. Then we published algal biorefinery and integrated approach, so published by Springer. Another book we published the microbial fuel cell that published by Springer. Now we are going to show you one video that is on 20-liter bioreactors for the continuous biohydrogen production by using Klebsiella the pneumonia at IIT Kharagpur, to give you some kind of idea on the biochemical process. And this is our website, and if you go to the website you will get the detailed information of our group, what kind of work we are doing. At the Indian Institute of Technology Kharagpur, we had a mission mode project on hydrogen production through biological routes. This is under the sponsorship of Eminari. And here we try to demonstrate that continuous hydrogen production by using immobilized whole cell by Klebsiella pneumonia, a local isolate. This is the bioreactor, and you can see this is the watch glass, and through the watch glass you can find out the volume of the liquid media present inside the reactor. And these are the other different units that is attached with this reactor. First is the feed tank, where you take the raw materials, and this is the peristaltic pump, and this is how the peristaltic pump is in operation. And you can see that this is the way how aseptically we can transport the feed from the feed tank to the reactor. Now here you can see there is a thermostat to show the temperature in the bioreactor, and here we have some sampling port from where we can draw the sample. This is the recycle tank, where we take the liquid which is coming out from the reactor and recycle back to the reactor, just to increase the retention time of the media, so that the percentage conversion efficiency of the substrate increases. Now this is kind of device we develop that is just to maintain some kind of vacuum in the reactor, and we have seen the Lee-Catlier principle, equilibrium constant equal to concentration of product divided by concentration of substrate. Now since our product is gas, we can easily remove if we apply some kind of vacuum, and then more substrate will convert it to product to maintain the equilibrium constant, and this is how our pressure regulating system works. Now we we open this pipeline so that the gas, whatever is accumulated, that can be collected in the gas collector. But this is passed through the carbon dioxide absorber. Here we use 50% of KOH solution to absorb carbon dioxide, and here we have a trap just to protect this KOH. It should not disturb our fermentation process. Finally, we try to find out that what is the nature of flame of the hydrogen. We find it is perfectly blue. So thank you very much. I wish you all the best, and I hope this course will be very much useful for you and for your future career. Thank you very much.